All right, thank you for having me. This is very exciting to give a talk on our work, especially so we are interested in all kinds of spectroscopy, microscopy techniques, especially vibrationally based. So I will talk about our efforts pushing STEAM and Raman scattering microscopy. So this is a nonlinear coherent Raman microscopy. I think this is something I started when I was a postdoc working with Sunny in 2008. I essentially carried over to in my Columbia lab. So let's see whether I can proceed. So very quick introduction of what STEAM and Raman is. This is actually a well established technique in the spectroscopy field. It's essentially a combination of Raman scattering with STEAM emission. So it was essentially discovered by physicists back in the 60s, but no one actually really thought about using it for imaging. So the first biological imaging paper was relatively speaking recent. So it's like 15 years ago. And then the entire field really pushed very hard on this. So I will give you a very quick introduction about the physics, and then the technology, and the very few simple examples. Typical examples of what we have done with that. So if you look at the energy diagram of STEAM Raman scattering, it's actually something rather straightforward. So you have a regular Raman, you use a pump laser, and then you wait for vacuum fluctuation, essentially, for the Stokes photon. But in STEAM Raman, instead of waiting for this vacuum fluctuation, we're inputting a strong laser beam. When the energy difference between the pump and the Stokes matches with the energy of vibration, this joint action will accelerate this rate by a huge factor. So experimentally, we measured under our microscopy condition, this rate of vibration transition can be accelerated up to 8 orders of magnitude. So, but this was an experimental observation. So, for a long time, since 2008, the field does not have a quantitative theory to explain why this is, 10 to the 8th and what factor contribute to that, and etc. So, in the past couple of years, so my group actually attempted a quite physics type of understanding. So we essentially right now we have a rigorous quantum mechanical theory to actually explain this and really understand what's going on in this. So I will give a very quick overview of this type of theoretical understanding. So the way to start this is actually by going back, to Einstein's A/B coefficients. So A is for spontaneous emission rate, and B is a rate coefficient for the stimulus emission. So this is well established as a foundation for light-matter interaction. If you think of Raman, it's actually quite analogous, because spontaneous Raman is just like spontaneous emission, but of course it's a scattering process. Whereas here, STEAM Raman is just like STEAM emission, again, it's a scattering process. So if you think about this way, you will say, maybe there's also something going on. There's some inherent, a deep connection between spontaneous Raman and STEAM Raman, something just like Einstein A/B coefficients. I think that's a correct physical intuition, but I have to say that no one has actually talked about this. This really, if you look at every literature or every textbook, there's no such a thing as A/B coefficients for Raman scattering. But if you think about it carefully, you think it should be, right? So essentially, starting from early this year, we start to think about how to actually do this. So just like Einstein A/B coefficient, you have to have A and B to start with. In other words, you have to quantify a rate process with some sort of a parameter, and then you try to figure out, whether you can connect those two parameters. Here, it's the same thing. You got to have a physical parameter to characterize Raman scattering, and then have another physical parameter to characterize STEAM Raman scattering, and then to see whether you can have an internal relationship. So, long story short, the way people used to characterize Raman scattering is Raman cross-section. Everybody knows this is well established, and every single Raman paper uses Raman scattering cross-section. But for steaming Raman, there's no such a thing. People still use Raman scattering for steaming Raman cross-section, which is in fact not quite right. So in this paper, we say, okay, the best way to do this, a logical way to do this, is actually make an analogy to two-photon absorption. Two-photon absorption use a two-photon absorption cross-section with a unit of so-called Gopper-Meyer. And we should use this type of steaming Raman cross-section to characterize steaming Raman process. So we can write a rate equation. The rate of steaming Raman should be the photon flux of pump, photon flux of Stokes, multiplied by what we say steaming Raman cross-section. So this will be the first step. We have to have a quantity, just like this will be like Einstein A coefficient. This will be like Einstein B coefficient. So we have to have this thing to start with. And then I think earlier this month, so basically in November, we published the paper showing that actually you can actually derive an equation, just like Einstein coefficient equation, to connect just two cross sections. So in the abstract, we say that we're inspired by AB coefficients, but now we can derive analogous equation for Raman cross section using an approach along the line of quantum electrodynamics. So I'm not going to show you the details, but just to show you the result. So this will be our equation. This is spontaneous Raman cross-section. This is a steaming Raman cross-section. They're connected by something like Einstein AB coefficient, but there's a difference here. There's a gamma. The gamma is the line width of Raman transition. So there's a slight difference. A slight difference because in Einstein's original derivation, he was doing it for broadband black body radiation. So the definition of the B is slightly different from the definition of our cross section. So this is actually more, I would say, this is more convenient and more physically grounded. So now we have equation for Raman and steaming Raman, just like Einstein's AB. Now with that, we can really write down a quantitative rate equation for steaming Raman microscopy. So the equation looks very similar. Very simple. This is the rate of SRS. This is the intensity of pump, intensity of Stokes. This is the number of molecules. This is Raman cross-section. Everybody uses that. Now we need to divide it by a term, which we now interpret this as a vacuum fluctuation. So effective intensity due to the virtual photons in the vacuum. So this is basically what we derived in this paper. Now we can put into use. And now once we have this rigorous quantitative rate equation, we can now predict how much faster this compared to spontaneous Raman. So we can calculate a so-called enhancement factor. So this theory will predict the enhancement factor of 2 times 10 to the 8th. And I told you that experimentally, this is actually recently measured to be 3 times 10 to the 8th. I think the difference is really probably because experiments could not really pin down some of the parameters. So we think the theory perfectly agrees with experiment. So now we have I would say we have a full quantum mechanical theory to understand, the whole processes at a very quantitative level. So with that, so this is how the microscopy works. I think this is all very established. What we use, we measure Raman gain, Raman loss, because when steaming Raman happens, there's energy flow between the laser field and molecule. The pump photon will lose. You have a relative intensity loss in the pump photon, and they have relative photon gain in the Stokes photon. So we can use modulation and locking amplifier to actually detect this relative intensity change. So it's like absorption measurements, but now it's a two beam, and then we use a modulation transfer to take a phase sensitive detection. So we can get down to short limit if the modulation is above megahertz. So that's one little pixel, and then we raster scan the sample. We will generate, just like a confocal, you scan across the sample, you will generate a 3-dimensional chemical map. And this has been commercialized by several microscope companies, such as Leica. They have this steaming Raman as one of their standard product. They're one of their high-end confocal microscopes. So I will tell you our efforts. So this was first, as I said, the instrumentation was first developed in 2008. The theory here was actually, we're still under review. So we now have a complete theory. And then I will tell you experimentally what we did in the past 10 years or so. So basically it's a sensitivity game, right? How we get from the originally, like millimolar concentration all the way to single molecule and super resolution. So there are a few technical key steps. First, we introduce the probes. And this, just like, fluorescence microscopy really benefit from the diversity of the fluorescent probes, we thought, we also need to develop our own vibrational probes. So, this is not for chemistry. This is too much of stuff to talk about it in this talk. But you can see here there are many different types. For example, for metabolites, drugs, functional sensors, photo switchable probes, for site-specific and multiplexing barcoding. There's all kinds of stuff. And I think I would say there's actually a real small field of its own developing Raman or infrared-based vibrational probes. And just a very quick example. So once we have this little probe, we can label, small molecules. This is one of our received paper in the field. So we can label pretty much any small molecules using this alkyne, and then we can see where the small molecules go. Because this group is much smaller than fluorescent fluorophores, we can actually really do a non-perturbative imaging of the small molecule metabolites, such as drugs. And we can recently move it to like little nanoparticles, because COVID vaccine is actually a nanoparticle. We actually make our own vibrationally labeled, deuterium labeled nanoparticles, and then this is what they look like. And then we inject to the immune system. We can see single nanoparticles, literally single nanoparticles in the immune cells. So this as a way to actually really study the specificity, the delivery, drug delivery, etc. So this is something that pharmaceutical companies are very interested in. And multiplexing, we all know fluorescence has very broad spectrum because of the dephasing. Now for vibrational, we can be very, very sharp. So here we engineered like 20 different vibrational probes. They all differ by structure a little bit, and they have very sharp peaks. And then we can use that to image, for example, 10 different organelles in live cells. This was not possible by any other means. Fluorescence can do 5 at most, but this is a live cell, so they're really constrained by their probes. But for our steaming Raman with vibrational probes, we can easily go up to 10, and we can do dynamics now. So that's the power of vibrational probes. And then another step is electronic pre-resonance. So far I'm not using electronic resonance yet, but you can do that. So you can see here, if your laser fixed here, if you just move your probe absorption from UV all the way to say close to your pump wavelengths, then the cross section will enhance drastically. This is a log scale, and this has been worked out by Albrecht. He has this famous theory, Albrecht resonance Raman theory. If you use their 8-term pre-resonance approximation, you can see that this curve is what he predicted. Use his equation, and that fit our data. And with this, we can actually really get to the micromolar, submicromolar concentration. So you can see here, this is a nitrile conjugated to a dye. Now you have a resonance. Then you look at the Raman of this electronic resonance mode, then the sensitivity can get down to submicromolar. Then we can see, turbulence. This kind of image can only be done by fluorescence in the literature. Now we're using antibody hook up with tubulin and antibody hook up with our dye, and then we look at Raman, then we can see this. So the beauty of that is that now we can actually really look at multiple colors. So I should have a slide here. So we can use this to look at, say, 20 different proteins simultaneously, because Raman peaks is 100 times narrower than fluorescence. The point is now we have the sensitivity and the spectral, resolution to look at this kind of stuff. Now here, steaming Raman excited fluorescence, that brings another 2 orders of magnitude down to single molecule. So the motivation is that since we're already using this type of resonance dyes, and the dyes all fluorescence to some extent, so why can we just combine Raman with fluorescence altogether? So the idea is, this is fluorescence, this is Raman. Now if we combine this into a single process, we call steam Raman excited fluorescence, then you will have vibrational, sharpness, you have vibrational selectivity together with fluorescence sensitivity, because fluorescence is background free now, but fluorescence doesn't have the chemical selectivity. Now Raman has it. So if you combine them, you are really combining the best of two worlds. So this actually has been long proposed in the literature by, for example, John Wright back in the 80s. He proposes, you look at here, right, steaming Raman and then fluorescence detection. He called this double resonance, but it doesn't work for him. So he's actually, he did experiments. If you look at a few years later, he did experiments, and then that shows that doesn't work. For example, you see, we report results that test the feasibility of double resonance excitation fluorescence by steam Raman. It was found the method was limited by a strong interfering two-photon process. Essentially, he didn't see the signal, and it was overwhelmed by the other background process. So essentially, the field doesn't really. So essentially, he proposed it, he killed it. Nothing happened for another 20, 30 years until Michel actually revisited this idea. So as you can see here now, we're in the modern times, and he's proposing you actually could do this possibly even for single molecule. So this is a proposal, okay? This is a proposal towards this, and I think the calculation shows it's actually promising. But I guess Michel told me that the experiments is probably too hard, and there are some technical details, but eventually there's really no experimental support for this proposal. So we went ahead and we realized one of the keys actually brings us to electronic pre-resonance. Because if you don't have any electronic pre-resonance, this Raman process is just too weak. So you will easily overwhelmed by other nonlinear processes. So we now know how to do this right. So we bring this to close to electronic resonance, but not quite yet. Therefore, we can gain, because you already have the electronic resonance anyway, right? You already have electronic state right here. So why don't you benefit from that? So we did that, and this is our apparatus. Pretty simple. So pump slopes, and the pump again serve as the third beam, and then we can do the steam Raman excited spectroscopy and imaging. And this is what things look like. We have one beam, second beam, and this thing serves as a third beam, and you will excite this electronic transition, and you will emit fluorescence. And we can see that if you tune the pump beam across a certain range, you will see the fluorescence will experience a sharp peak exactly at the vibrational position as you expected. So this is really a Raman transition by nature, but it's detected by fluorescence, and the signal can be very high. So we can get down to nanomolar now, and you can show we can indeed do single molecule. So this is a room temperature single molecule. We scan across the vibrational peak, we can see it will show up right in the right position. So this is really a demonstration of single molecule vibrational spectroscopy without any surface enhancement. Because with surface enhancement, you can do a lot of things, but this is without surface enhancement. So I think this broadens a lot of biophysical studies where it's difficult to put nanoparticles close to the molecules. So we even did isotope. We use different isotope doping to show how we're gonna fine-tune the peak. So if you look at absorption fluorescence, isotope doesn't change it as we expected, but you can see this in the steaming Raman excited fluorescence. In steaming Raman excited fluorescence, you can see different isotopes. Edited version will have different peaks. So again, proving we are looking at Raman transition through fluorescence. So that's a single molecule. And finally, we can push to super resolution because it's almost obvious, because once we have a fluorescence hooked up in your process, you can just harness all the fluorescence-based super resolution techniques. Nowadays, everybody understands that now. So you can use single molecule, pump, storm, you can use STED. So we actually took a simple approach. We just use STED. So you use steam emission depletion. And the way we're doing this is that, when the molecule is right here, you can use a beam, a STED beam, a donut beam to squeeze the point square function. Of course, you have to delay for a few picoseconds for the vibration relaxation to come down. But, in principle, there's really no issues with that, completely feasible, from a physical point of view. So we build apparatus, and you can see here, this is a signature curve the remaining signal as a function of step beam power undergoes this nonlinear saturation. So that's a signature curve, shows that you can do step. So we haven't pushed it for too far because the molecules wasn't so bright. So we really don't have a, I mean, in this paper, we didn't have a right molecule. So the fluorescent quantum yield is only a couple percent. So we're limited by essentially by the brightness of the dye. But you can see the physics stuff does work. We have a factor of 2 improvement in this simple demonstration. We can move it even to biological samples. So you can see here, with and without stat beams, resolution improvements about factor of 2. Again, we are working on improving our dyes in terms of the quantum yield. So with say 10 of 50 times better quantum yield, this resolution should be a lot more better. I mean, as you know, according to Stephen Hell, the stat microscopy is only limited by the photophysics. So if we have a good photophysics of the dye, we can keep pushing the resolution. So with that, I want to I think this is a good time to stop. Maybe, yeah. So I just want to thank my group who actually made a contribution, and we are also actively recruiting graduate student postdocs. Thank you. I would like to take any questions you might have.
Thank you very much for the exciting talk. Very exciting methods. Maybe there are questions for the talk. Some quick ones.
I saw Michelle.
Yes, please go ahead, Michelle.
Yeah, thanks a lot, Wei. Very exciting and very beautiful. I congratulate you, for this dream experiment that we want to do already more than 20 years, and you made it. It's great. Thanks. You mentioned that you could only make it because you need the pre-resonance Raman condition. But I have a simple question there. I mean, wouldn't it be possible to compensate this pre-resonance by putting more power on the pump and stokes?
I mean, yes. I mean, yes. I mean, there's one way is that we are using oscillators. So we're using 80 megahertz beams. So in that sense, our peak power is not terribly high. And we are moving towards a lower rapid rate laser, for example, megahertz. And therefore, the peak power will be 100 times higher. So it's possible that if we move to the megahertz excitation laser, we probably could relax the requirement on pre-resonance. Therefore, we can be more flexible. So at that time, I would say 5 years ago, we got to rely on some sort of a resonance enhancement.
Okay, so maybe in the future.
Yeah, I agree with you. There's no
because it's many conditions. So the more general you make the method, the more powerful it becomes.
Yeah, I agree. Generally, there's no fundamental requirement. It's just that
exactly.
practical reasons.
Great. Okay. Thanks a lot.
The second question by Randy.
Yeah, beautiful work, Wei. My question is about the first story, about the connection between the stimulated, about how you determine the stimulated Raman cross section. So, most people who are doing SRS, right, are using pulse lasers. It makes sense. They're taking advantage of the high peak powers. There's a small community of people doing SRS with CW lasers, and so I feel like the general knock on that is that, well, you don't have higher peak powers, so you lose sensitivity. But it seems like maybe your equation suggests that maybe you get some back because you're using a more narrow excitation. Is that correct? Is there sort of a do you get kind of apparent larger cross sections when you do CW stimulated Raman?
Well, okay. So a good point. It's so basically when we say pulse laser gains, okay, we say pulse laser give a more sensitivity to some limit. Basically, meaning if you have picosecond pulse, you're still gaining. But if you go all the way to femtosecond pulses, then you no longer gain anymore because you are essentially broadening your pulse. You are no longer gaining that sharp resonance. So in that sense, CW. So in my view, I mean, in these equations, CW is essentially the cross section is about the same between CW and the picosecond lasers. So in that region, you should use picosecond, you should use higher peak power. And once you go beyond picosecond pulses, then there's another story, because then you're broadening your pulse, your effective cross-section will go down.
Got it. Thank you.